Hello, dear students. In the last period, we have studied the examples of enzyme catalysis. We are going to study some more examples. So, the sixth example is in stomach, the pepsin enzyme converts proteins into peptides, while in intestine, the pancreatic trypsin converts proteins into amino acids by hydrolysis. Seventh example, conversion of milk into curd. It is an enzymatic reaction brought about by the lactobacilli enzyme present in the curd. So if you could notice, most of the name of the enzymes comes from types of reaction or from reactant by adding suffix ase. Let us revise all the enzymes that we have studied. See, we have different enzymes and their sources. So this invertase enzyme comes from yeast, zymase comes from yeast, diastase comes from malt, maltase comes from yeast, urease comes from soybean, and pepsin comes from the wall of the stomach. Now, what are the various enzymatic reactions that are affected by these enzymes? So, invertase turns sucrose into glucose and fructose. You can see that sucrose by inverted is it's converted into glucose and fructose. Same way, zymase converts glucose into ethyl alcohol and carbon dioxide. Diastase converts starch into maltose. Maltase converts maltose into glucose. Urease converts urea into amino acid and carbon dioxide. Urease converts urea into ammonia and carbon dioxide. Proteins, pepsin in the stomach convert proteins into amino acids. So this is how we can understand about the various enzymes. Enzymes are very specific and can be specifically used for biochemical reaction, cannot be used for another reaction. So once they are used in for a particular reaction, they cannot be used for another reaction, needed in a very small quantity. So enzymes are always needed in a very small quantity. I have shared some of the characteristics of enzyme catalysis. Now we are going to study the characteristics of enzyme catalysis in detail. Enzyme catalysis is a unique in its efficiency and high degree of specificity. The following characteristics are exhibited by the enzyme catalysis. Number one, most highly efficient. One molecule of enzyme may transform 1 million molecules of reactant per minute. It shows that enzymes are very effective, highly efficient, highly specific in nature. Each enzyme is specific for given reaction. It means one catalyst cannot catalyze more than one reaction. Same way, one enzyme cannot catalyze more than one reaction. For example, enzyme urease catalyzes the hydrolysis of urea only. It does not catalyze the hydrolysis of any other amide other than the urea. The third characteristic is highly active under optimum temperature. So we should understand first what is optimum temperature. So let us understand or let us define what is optimum temperature. The rate of enzyme catalysis reaction become maximum at a definite temperature called the optimum temperature. If temperature is more than optimum temperature or less than the optimum temperature, the ability to catalyze the biochemical reaction for enzyme decreases. Optimum temperature range for enzyme catalysis is 298 to 310 Kelvin. Human body temperature is 310 Kelvin, which is suitable for the enzyme catalytic reactions. Fourth characteristic is highly active under optimum pH. The rate of an enzyme catalyzed reaction is maximum at particular pH called optimum pH. Optimum pH range for enzyme catalyzed reaction is between 5 pH to 7 pH. So this is how we can understand a few of the very important characteristics of the enzyme catalysis. Now we have the fifth characteristics that we should understand. Increasing activity in the presence of activators and coenzymes. So first we should understand what is coenzyme. The enzymatic activity is increased in the presence of certain substances called coenzymes. Coenzymes are non-proteinous substances like vitamins. Coenzymes remains present along the enzyme. Coenzyme enhances activity of enzymes considerably. So these are the characteristics of coenzymes. Then we have the activators. Activators are metal ions such as Na plus 1, Mn plus 2, Co plus 2 and Cu plus 2, etc. These metal ions, when weakly bonded to enzyme molecules, increases the catalytic activity of an enzyme. Let us understand the same with the example. There is an enzyme named is amylase. Amylase, in the presence of sodium chloride means it has Na plus ions. So amylase in the presence of Na plus ions are catalytically very active. Then we have the sixth characteristic, that is the influence of inhibitors and poisons. First, we should understand the definition. Substances that inhibits or decreases the ability of enzymes are called enzyme inhibitors or enzyme poisons. Like ordinary catalyst, activity of enzymes even influenced by inhibitors and poisons. Now, how do they act? How do these inhibitors or poisons act? Actually, the inhibitors or poisons interact with the active functional group on the enzyme surface and often reduce or completely destroy the catalytic activity of enzymes. So they either reduce or they destroy the catalytic activity of the enzyme by working on the activating functional group present on them. We can understand it with the example. Many drugs in our body acts like a enzyme inhibitors and they function against the diseases. So this is how we can understand the various characteristics of the enzymes. Now we shall study the mechanism of enzyme catalysis, that we'll study into the next video. Thank you. I hope you are enjoying. Happy learning.